So typically, when we think of a disease, there's one causative agent. In the case of periodontal disease, it's actually more likely caused by several bacteria that come together and share resources. We're trying to identify unique proteins that are upregulated when these bacteria come together. And we're also looking at it from a top-down approach of seeing if we can find chemicals, find therapeutics, find things that will actually scrub the biofilms away from this subset of bacteria, potentially leaving the more healthy, beneficial bacteria behind. We were looking at an operon, a collection of genes in a little system that will do a particular function. The gene products are responsible for the recognition and degradation of a link of repeating chains that is sulfated. And the gene products, when they're converted into proteins, they're able to recognize that chondroitin sulfate, break it down into simpler components. It not only destabilizes the teeth, but bacteria gain resources. The level of resolution, the level of detail that we get from the synchrotron is really unparalleled, and it allows us to really definitively, or with a large probability, a large likelihood, say how these things look. So it's kind of glimpsing, not under a microscope, but under a super microscope in order to really see what these proteins look like. The Canadian light source has been really a cornerstone for our research right from when my lab started. We don't have a home X-ray source and detector, meaning that we can't evaluate our samples appropriately. So the capacity to send it to Saskatchewan, we've had amazing support both in training and as well for data interpretation, analysis, and recovery that has allowed our research to proceed. Otherwise, really, we wouldn't have been able to characterize the two structures that we presented in this paper.